Assalamu alaikum. Good morning everyone. Today we are going to talk about the physiology of bile secretion and the functions of the gallbladder. The objectives of today's lecture are to describe how bile is formed by the hepatocytes in the liver, trace the path of bile through the bile canaliculi to either the duodenum or the gallbladder depending on whether we are in the fed state or the interdigestive state, enumerate the organic constituents of the bile and appreciate that the chief inorganic constituents are sodium chloride and sodium bicarbonate, describe the neural and hormonal factors that regulate the emptying of the gallbladder and the relaxation of sphincter of Oddi, and explain the functions of the gallbladder, and finally recognize the function of the bile salts in fat digestion and absorption. As you know, the liver is a solid pyramid-shaped organ enclosed by peritoneum. It is the largest internal and most important metabolic organ in the human body, weighing about 1.4 kg to 1.6 kg in a healthy adult. It represents about 2.5% of the total body weight. The liver is a highly vascular organ. It receives about 25 or 1/4 of the resting cardiac output. As you can see here, the liver has a dual blood supply. It receives oxygenated blood from the aorta through the hepatic artery, and it receives venous blood, deoxygenated venous blood that is rich in nutrients through the hepatic portal vein. As I said, the liver has dual blood supply. The portal vein that brings about 80% of the hepatic blood supply or the liver blood supply. It carries blood from the gut, which is rich in nutrients, pancreas and spleen. And we have the hepatic artery that supplies about 20% of blood to the liver. It arises from the celiac axis, which is a branch of the abdominal aorta, and ensures that the liver is well perfused and supplied with oxygen for metabolism. As you can see here, the liver has dual blood supply, oxygenated blood through the hepatic artery, and deoxygenated blood that is rich in nutrients through the portal vein. The blood will mix in the liver or hepatic sinusoids, then it will drain through the central vein into the hepatic vein, and then inferior vena cava, and then the right atrium of the heart. What are the different functions of the liver? The overall functions of the liver are: number one, secretion of bile salts, which is the topic of today's lecture, which aid in the digestion of fat, in the digestion and absorption of fat. Excretion of cholesterol and bilirubin. These are derived from the destruction of worn out red blood cells or aged red blood cells. Metabolic processing of the major categories of nutrients. These are carbohydrates, proteins and lipids. Detoxification or degradation of body wastes and hormones, as well as drugs and other foreign compounds. Number 5, synthesis of plasma proteins, including different clotting factors and transport proteins for steroid and thyroid hormones, as well as cholesterol. Number 6, storage of glycogen, fats, iron, copper, and many vitamins like vitamin A, D, K, and B12. Then the activation of vitamin D in conjunction with the kidney. Both the liver and kidney are important for the activation of vitamin K. Finally, the removal of bacteria and worn out red blood cells by the fixed tissue macrophages that we call them the Kupffer cells in the liver. Today, our topic is mainly focusing on the secretion of bile salts and the excretion of cholesterol and bilirubin. Bile serves two important functions, and these are: it plays an important role in fat digestion and absorption by the following: emulsifying the large fat globules of food into minute fat droplets, as we can see here, we have a large fat globule, and with the help of bile salts, this fat globule is separated or divided into smaller fat droplets, and the bile salts will prevent the fat droplets from recoalescence or from forming a large globule again. Another important function of bile is that they contribute to the formation of micelles that aid in the absorption of the digested fat end products through the intestinal mucosal membrane. Another important function also is the bile serves as a mean for the excretion of waste products from the blood, and these waste products include bilirubin, which is the end product of hemoglobin destruction, and cholesterol. Bile is the only route by which the body can dispose of cholesterol in its native form or following its conversion to bile acids. So either cholesterol is released as cholesterol per se, or it is converted to bile acids and then released as bile acids into the bile. Bile that is formed by hepatocytes is secreted into the bile canaliculi, and then from the canaliculi it flows into the bile ductules. So the bile that is formed by the hepatocytes is released into the bile canaliculi, then finally this will flow into bile ductules, then into bile ducts in the portal tracts at the periphery of the liver or hepatic lobules. Then the bile will flow through the intrahepatic ducts and then to the right and left hepatic ducts. From there, the two hepatic ducts, the right and left ducts, will join to form the common hepatic duct, as we can see here in this illustration. They form the common hepatic duct that will be joined by the cystic duct to form the common bile duct, and then the common bile duct will be joined by the pancreatic duct at the ampulla of water or the hepatopancreatic ampulla, and to release the secretions into the duodenum, and these ducts are controlled by the sphincter of Oddi. Bile is continuously secreted by the liver, independent of the fed state. During the interdigestive period, the bile is diverted to the gallbladder because the sphincter of Oddi is closed. So in the gallbladder, the bile will be stored and concentrated. During the fed state, it enters the duodenum because the sphincter of Oddi is open. So the bile will enter the duodenum as a result of the combined effects of gallbladder contraction or emptying and the increased bile secretion by the liver. The volume of bile that is released daily is between 600 to 1200 milliliter in an adult male, depending on the degree of stimulation. The bile has a golden yellow color. It has an alkaline pH ranging between 7.5 to 8.05, and it is isotonic compared to the plasma. As you can see here, the bile is composed mainly of water. It also contains bile acids, cholesterol, phosphatidylcholine or lecithin, bilirubin, fatty acids, and inorganic ions. As you can notice that the ratio between bile acids to cholesterol and lecithin is 10 to 1 to 0.3, and this ratio is very important to maintain the solubility of the different components of bile. In human, there are two types of bile acids. These are the primary bile acids and the secondary bile acids. The primary bile acids are derived from cholesterol, as you can see here in this illustration, in the liver, and then when the primary bile acids flow into the intestinal lumen, they will be acted upon by the intestinal bacteria or the microbiota in the intestine to be converted to secondary bile acids. Primary bile acids and secondary bile acids can be conjugated or unconjugated. First, let's know the two main types of primary unconjugated primary bile acids. These are cholic acid and chenodeoxycholic acid. The secondary bile acids, the unconjugated forms, we have two main forms, deoxycholic acid and lithocholic acid. Primary bile acids, cholic acid and chenodeoxycholic acid, are generated directly from cholesterol in hepatocytes, the liver cells. The secondary bile acids, deoxycholic acid and lithocholic acid, are generated by the action of intestinal bacteria on primary bile acids in the intestinal lumen. Unconjugated primary and secondary bile acids can be converted to conjugated bile acids when they are conjugated with the amino acid glycine or taurine in hepatocytes to form the conjugated bile acids. The conjugated bile acids are more easily or are more prone to ionization, so they ionize more easily, so they are more water soluble. Bile acids are amphipathic, which means they have both hydrophilic and hydrophobic domains. At neutral pH, the bile acids are more easily or mostly ionized and are referred to as bile salts. Conjugated bile acids, as I said in the previous slide, ionize more easily or readily than the unconjugated bile acids and thus usually exist as salts of various cations or positively charged ions, example sodium glycocholate. Bile salts are much more polar than bile acids and have greater difficulty penetrating cell membranes, which keep them inside the lumen for longer time to perform their function. Consequently, the small intestine absorbs bile salts much more poorly compared to bile acids. This property of bile salts is important. The small intestine or the ileum will absorb bile salts only after all the lipids have been absorbed. The total bile secretion by the liver is composed of an initial portion that is secreted by the hepatocytes into the minute bile canaliculi and contains large amount of bile acids, cholesterol and other organic constituents. It is secreted into the bile canaliculi between the hepatic cells, as we saw in one of the previous slides. A second portion of secretion is added to the initial bile as it flows through the bile ducts. This secretion is a watery solution of sodium and bicarbonate secreted by the secretory epithelial cells or the cholangiocytes that line the bile ductules and ducts. This second secretion can increase the total quantity of bile by an additional 100%. This secretion can double the amount of secretion of bile that is originally secreted by the hepatocytes into the bile canaliculi. The second secretion is stimulated especially by secretin, by the hormone secretin, which causes the release of additional quantities of bicarbonate to supplement bicarbonate in the pancreatic secretion. And all these bicarbonate ions are meant for neutralizing acids that empty into the duodenum from the stomach. Next, the bile flows into the biliary tree and either empties directly into the duodenum or is diverted for minutes up to several hours through the cystic duct to the gallbladder, depending on the fed state of the person. Now let's talk about the gallbladder. The gallbladder is a sac or membranous or muscular sac that is present underneath the liver. It has a duct that joins the common hepatic duct to form the common bile duct that will empty the secretion into the duodenum through the ampulla of water. The gallbladder has several important functions, and these include the storage, concentration, and discharge of bile into the intestine. Bile is normally concentrated about 5 times and can be concentrated up to a maximum of 20 folds or 20 times. Bile leaving the liver in the hepatic duct between meals or during the interdigestive state is diverted into the gallbladder as a consequence of the closure of the sphincter of Oddi. The gallbladder musculature relaxes to receive this amount of bile. Hepatic biliary secretion between meals is about 450 ml. However, the human gallbladder can accommodate to hold about 20 to 50 ml of bile. The volume of bile should be reduced and concentrated from 450 to a maximum of 50 ml during the interdigestive state to be accommodated by the relaxing gallbladder. The gallbladder is able to concentrate bile because it has the ability to reabsorb sodium, chloride, and water. As you can see here in this illustration, we have the cholangiocytes that line the gallbladder, or these are the mucosal cells, and we have here the bile in the lumen, and we have the interstitial fluid. We can see that the cholangiocytes can reabsorb sodium, chloride, and water and move them to the interstitial fluid. Sodium and chloride ions in the gallbladder lumen are exchanged for hydrogen and bicarbonate respectively from the cytoplasm of the epithelial cells or cholangiocytes that line the gallbladder. The sodium ions are removed from the epithelial cells along the basolateral border by the sodium-potassium pump, and chloride ions will exit the cell via the chloride channels in the basolateral membrane. The accumulation of sodium chloride in the intercellular space will create an osmotic gradient for the transfer or the movement of water from the lumen of the gallbladder into the interstitial space. Water passes from the lumen through the channels in the membranes and between the cells along this gradient. As you know that we have channels in this plasma membrane that are or that allow water to pass, and we call them aquaporin. The overall result of this reabsorption of sodium, chloride, and water is that many components of gallbladder bile are more concentrated compared with the hepatic bile, which is the bile before entering the gallbladder and before being concentrated. The bile remains isotonic to plasma because the micelle concentration increases. The aggregates of bile salts, phospholipids, and cholesterol in the micelles have only a small osmotic effect. So the concentration of the bile will be isotonic because the micelles become more concentrated with the bile salts, phospholipids, and cholesterol, and as I said, this will have very small osmotic activity. The protons secreted by the gallbladder epithelium here, as we can see by the sodium hydrogen exchanger, will decrease the bile pH. A decrease in the bile pH will increase the solubility of calcium salts, and this is important in reducing the possibility of gallbladder stone formation, which is a common problem, especially when there is inflammation in the gallbladder. Epithelial cells secrete also mucus to protect their apical membrane from the damaging effect of bile salts. Excessive mucus secretion may contribute to gallstone formation. Here we can see the difference between gallbladder bile and hepatic bile. We can see clearly that the gallbladder bile is different from the hepatic bile. As you can see, water content of the hepatic bile, of the gallbladder bile, is less compared to the hepatic bile. Bile salts are about 5 to 6 times more concentrated compared to the hepatic bile. Bilirubin is about, again, 9 times more concentrated in the gallbladder bile compared to hepatic bile. The same for cholesterol. Fatty acids are also more concentrated. Phospholipids are more concentrated, and we can see also the ions are different because of the reabsorption of sodium and chloride. Their amount in the gallbladder bile are smaller compared to the hepatic bile. Now let's talk about the control of bile formation and bile release. Bile formation from the hepatocytes and the cholangiocytes that line the bile ductules and bile ducts, and the release of concentrated bile from the gallbladder. As I said, bile is formed by hepatocytes, and also an addition is released from cholangiocytes lining the hepatic ductules and ducts. So let's talk about the control of bile formation. Bile formation is controlled in different ways. The canalicular bile, where the major determinant of bile acid secretion is the concentration of bile acids in the portal or hepatic portal vein. The larger the concentration, or the higher the concentration of bile acids in the hepatic portal blood, will lead to larger amount of bile acids released by hepatocytes into the canaliculi. And that concentration of bile acids in the hepatic portal blood depends on the enterohepatic circulation of bile acids that we'll cover later on in this lecture. On the other hand, the control of ductular bile formation is mainly under the effect of the hormone secretin that stimulates the production of bicarbonate-rich secretion that is potentiated by the hormone cholecystokinin. Also, ductular bile formation is under the control of vagal nerves that release the neurotransmitter acetylcholine, which in turn stimulates ductular bile secretion, but this effect is weak compared with the hormone effect. Now let's talk about the control of bile release from the gallbladder, what we call gallbladder emptying. Gallbladder emptying is mainly the function of the hormone cholecystokinin. Cholecystokinin causes smooth muscle contraction in the gallbladder wall and the relaxation of the sphincter of Oddi, and this will allow the concentrated bile to flow into the small intestine. Another controller is the parasympathetic or vagal stimulation that results in the contraction of the gallbladder and relaxation of the sphincter of Oddi. Again, this effect is relatively weak compared to the hormonal effect or the effect of cholecystokinin on gallbladder emptying. On the other hand, sympathetic stimulation will result in reduced bile secretion and the relaxation of the gallbladder. We know very well that the sympathetic system, or the sympathetic division of the autonomic nervous system, has an overall inhibitory effect on the digestive system. So to summarize here, we can see that bile acids via the blood in the hepatic portal vein will stimulate parenchymal secretion because they will be taken up by hepatocytes on one side and released from the opposite side of the cell into the bile canaliculi. On the other hand, secretin via the bloodstream will stimulate the liver ductal secretion, and this will add to the bile that is released by the hepatocytes. For the gallbladder, the bile will be stored and concentrated up to 15 times in the gallbladder. Then it will be potently, or the gallbladder emptying will be potently stimulated by cholecystokinin that stimulates gallbladder contraction and relaxation of sphincter of Oddi. As I said, vagal or parasympathetic stimulation has a weak effect on gallbladder contraction. Here we can see that how acidic chyme or chyme that is rich in protein, digestion products and fat can affect bile secretion. Acidic chyme in the duodenum will stimulate the release of secretin from the S cells in the duodenal glands, and this will increase the plasma secretin level, which will flow via the blood to the liver, and this will stimulate bile secretion from the cholangiocytes lining the bile ductules and ducts. Protein digestion products and fat in the duodenum will stimulate the CCK cells to release cholecystokinin. Its level will increase in the plasma or in the blood. Cholecystokinin will stimulate gallbladder contraction and the relaxation of sphincter of Oddi. All these effects will release bile into the duodenum, and this will lead to the neutralization of acid because bile is rich in bicarbonate, and the emulsification of fats because bile is rich in bile acids. As I said earlier, the bile salts are amphipathic. They have hydrophilic and hydrophobic domain. So they have two domains, and this feature helps them to break down large fat globules into smaller fat droplets, and this will increase the surface area of the fat droplets, and this will help in the digestion of the fat by the pancreatic lipase that is released with the pancreatic juice. Therefore, bile salts help break down of fat globules by reducing their surface tension in the duodenum into tiny droplets, and this process is called emulsification, which provides a larger surface area on which the enzyme pancreatic lipase can act to digest the fats into fatty acids and monoglycerides. As we can see here, at stomach will release the chyme that contains large fat droplets. This will mix with the bile salts that is present in bile and will result in emulsification of these large fat droplets into fat globules into small fat droplets, and this will increase the surface area, as I said, for the action of the pancreatic lipase to digest the triglycerides into free fatty acids and monoglycerides. The monoglycerides and the free fatty acids that are released by the action of the pancreatic lipase on the small fat droplets will enter into micelles. The micelles are composed of bile salts, the phospholipid lecithin, and cholesterol, as you can see here. This is the structure of the micelle. These are the bile salts molecules, and we have the lipids inside the free fatty acids and the monoglycerides, and we have the lecithin here forming the outer shell of the micelle. So this micelle will make those those free fatty acids and monoglycerides water soluble and will allow carrying them to the absorptive enterocytes in preparation for their absorption. The micelles act as a transport medium to carry the monoglycerides and free fatty acids to the brush border of the intestinal epithelial cells, as we can see here. This is fat droplets. The pancreatic lipase is acting on its surface, releasing free fatty acids and monoglycerides that will enter the micelle, and then the micelle will move to approach the absorptive surface of the brush border. Now let's discuss the enterohepatic circulation of bile salts. Why we need that enterohepatic circulation? Because our body contains a certain total amount of bile acids. We call the total bile acid pool. And every day, to have efficient lipid absorption, we need a certain amount of bile acids. However, the amount that we need for the absorption of all lipid that is present in our meal is much higher than the total amount of bile acids that is present in our body. So the body should recycle that bile acid with each meal several times to achieve the amount needed for the reabsorption. So the enterohepatic circulation of bile salts is the recycling of bile salts between the small and large intestine and the liver. For efficient fat digestion and absorption, we need 15 to 30 grams of bile acids per day. The total amount of bile acids in the body, primary, secondary, conjugated or unconjugated at any time, is defined as the total bile acid pool. In a healthy person, the total bile acid pool ranges between 2 and 4 grams. This is much lower than the amount we need to have efficient bile for efficient lipid absorption. So because the hepatocytes do not have the capacity to produce new quantity of bile salts needed to aid in the digestion of fat in each day's meal, the enterohepatic circulation of bile acids in this pool is physiologically extremely important. By recycling several times during a meal, a relatively small bile acid pool can provide the body with sufficient amounts of bile salts to promote lipid absorption. In a light eater, the bile acid pool may circulate 3 to 5 times a day. In a heavy eater, on the other hand, it may circulate 14 to 16 times a day. So that total bile acid pool can circulate up to 16 times per day in heavy eaters. And this is an illustration that shows how the enterohepatic circulation happens. As you can see, we have the bile salts produced from cholesterol by the hepatocytes. They will be conjugated. They are either primary or secondary, and then they will be released with the bile concentrated in the gallbladder if we are in the interdigestive state, and then released in the small intestine or duodenum as conjugated biles. They will perform their function as in emulsification and formation of micelles, and after they finish their function, they will be reabsorbed. So they will be reabsorbed along the length of the terminal ileum and as well as the large intestine. We can see that reabsorbed bile acids and salts will go back to the liver. They will be taken up by hepatocytes and excreted in the bile. As you can see, bile, as it flows through the small intestine and large intestine, they become deconjugated. They might be changed into secondary bile acids due to the action of the microbiota, but all of them can be reabsorbed and returned back to the liver. Inflammation of the ileum can lead to bile salt malabsorption and results in the loss of large quantities of bile salts in the feces or in the fecal material. And as we lose that bile acid pool, there will be malabsorption of fat in the inflammatory bowel disease patient, because this will lead to dramatic decrease in the total bile acid pool in the body. So the amount that is involved in enterohepatic circulation will be much less. So the enterocytes along the length of most of the intestine are impermeable to conjugated bile salts. This effect maintains a high concentration of bile salts to promote fat digestion and absorption, which is usually complete before the terminal ileum is reached by the chyme. The enterocytes of the terminal ileum possess or have transport mechanism for bile salts, which is sodium-dependent bile salt transporter that is present in the brush border of the enterocytes, and these transporters preferentially absorb the negatively charged conjugated bile salts. Bile salts are also metabolized by intestinal bacteria or the microbiota. This can involve deconjugation which increases their lipid solubility and reabsorption. In addition, bacteria are responsible for the conversion of primary bile acids to secondary bile acids, which may be absorbed too. The unconjugated bile salts may be passively absorbed, and this accounts for about 10% of the recycling of bile salts if they become protonated, which removes their negative charge and make them more lipid soluble. The bile salts are transported in the hepatic portal vein to the liver bound to albumin because they are water insoluble, where they are again extracted and secreted into the bile. Bile salts that escape absorption from the small intestine are excreted in the feces, and this is approximately 5% of the bile salts entering the duodenum each day, which is about 0.5 gram per day. The liver synthesizes this quantity each day, which is about 0.5 gram per day, to make up for the loss. Increases in bile salt excretion are matched by increases in the synthesis in order to maintain the total bile salt pool in the body. And with this slide, we end our lecture today. And these are the references that were used to prepare this lecture. Thank you very much for your patience, and I hope that you got some benefit, and all questions are welcome via email. Thank you. Please stay safe and have a nice day.